And now we are going to our another presentation. It's the one of the APMAS Congress pre-recording presentation is going to be here. Professor Mina Raileanu is going to present her presentation about antimicrobial peptides as a new combination agent in cancer therapeutics. She's from Horia, Hulubei National Institute for R&D in Physics and Nuclear Energy Engineering from Romania.
My name is Raileanu Mina, and today I would like to present to you my results regarding antimicrobial peptides used as a new combination agent in cancer therapeutics. We will start with a short introduction in which I will describe the type of cell culture we will use and the antimicrobial peptides. So, cells in the physiological environment have constant interaction with the extracellular matrix, regulating complex biological functions like cellular migration, apoptosis, transcriptional regulation, and receptor expression. In vitro experimental data cannot be translated into clinical trials completely when cells are grown in bidimensional conditions or 2D conditions, since complicated cellular signals between cells and its matrix cannot be reproduced. Three-dimensional cell cultures address this challenge and serve as a better model representing in vivo physiological conditions closely. So with three-dimensional cell cultures, we could study the invasive character in the case of tumors and/or physiological characteristics of cellular aggregates. The other novelty used in our study are the antimicrobial peptides, or AMPs, also known as host defense peptides. They exist in almost all species of vertebrates, invertebrates, and plants. Most AMPs consist of less than 100 amino acid residues, and they share several common features including cationicity, hydrophobicity, and amphipathic structure. The antimicrobial functions of cationic AMPs can be categorized into two major mechanisms. 1. Disrupting microbial membrane integrity via interactions with negatively charged components. And 2. Inhibiting the function and synthesis of intracellular DNA, RNA, and protein. In addition to their direct antimicrobial activity, AMPs have immunomodulatory properties such as chemotactic activity to immunocells and lipopolysaccharides neutralizing ability. The characteristics these peptides have give them an increased affinity for cell membranes. Compared to the healthy cell membranes, cancer cell membranes are negatively charged, facilitating the attachment of the peptide to the membrane, followed by its disruption through different mechanisms. Moreover, AMPs have a reduced toxicity to rapid proliferating healthy cells. First, we have to see the differences between 2D and 3D culture, and so we have a comparison. The most important difference between these two types of culture is that 2D cultured cells do not mimic the natural structures of tissues or tumors. In this culture method, cell-to-cell and cell-to extracellular environment interactions are not represented as they would be in the tumor mass. These interactions are responsible for cell differentiation, proliferation, vitality, expression of genes and proteins, responsiveness to stimuli, drug metabolism, and other cellular functions. The changed morphology of the cells can affect their function, the organization of the structures inside the cell, secretion, and cell signaling. Due to the disturbances in interactions with the external environment, cells growing adherently lose their polarity, which changes the response of those cells to various phenomena, such as apoptosis. These disadvantages are not met by using the 3D cell culture. This type of cell culture being much closer to a real tumor environment. So we can consider the spheroid or the 3D cell culture method the missing link between the 2D cell culture and animal testing. Other differences between these type of cell cultures are that in 2D cell culture, cells don't adhere properly. In 3D, cells attach one to one another and form cell-to-cell tight bonds. Also, gap junctions between cells are extremely rare in 2D cultures. In 3D, gap junctions are common, having an important role in ions, small molecules, and electric impulse exchange. Little to no tissue differentiation we can see in 2D cell culture. Also in 3D, cells have an accentuated differentiation and adherence. And last but not least, cells can be easily damaged by small concentration of medicine in 2D cell culture. In 3D, cells are not easily damaged by chemotherapy compounds in small concentration or by minimal doses of radiation. And so we can see in the mechanical process of cellular spheroid formation, there are two important stages, cellular aggregation and spheroid compaction. In the cellular aggregation phase, we have the forming of interactions between integrin and extracellular matrix on the surface of the cells. On the spheroid compactions, we have cadherin, which is another protein of ligament protein on the surface of the cells. And we can see that in the first stage we have an expression and accumulation of cadherin on the surface of cells. And in the last stage we can see the formation of cadherin-cadherin interaction, bringing the spheroid together and being more compact. The most important characteristic of the spheroid is the three layers which consists the spheroid. We have the external layer, which is called the proliferative zone, which is formed out of healthy cells because they have free access to the nutrients in the medium and gases like oxygen. And after that, we have the quiescent zone, in which the cells have divots and dents in the membrane and also a smaller nucleus. And in the center, we have the necrotic zone in which the toxins and other compounds are gathered, in which the cells are practically in apoptotic and necrotic state because of the lack of access to the nutrient, oxygen, growth factors, or proliferating factors. Our main goal that we wish to achieve was to obtain and characterize spheroids, their evolution in time, morphology, also studying the anti-tumoral effects of the two compounds that have different mechanisms of action. We started with the colorectal adenocarcinoma cell line, to be more exact, it's HT29. We've grown it in a 2D cell culture till confluence, and then we trypsinized it and formed spheroids out of it with the help of rounded bottom cell plates. We waited till the spheroids were formed and then applied the treatment with doxorubicin, which is a classical chemical compound using chemotherapy in different concentration, and gramicidin A, which is our antimicrobial peptide. After the treatment, we watched the evolution in time and structural changes, the fluorescence of doxorubicin and its penetration in spheroids, and as viability test, we watched the mitochondrial activity through MTT and the energy levels inside the cell through ATP tests. The first step in our experiment was to watch the evolution in time and also choose cellular density. And so we seeded different cellular densities from 1,000 to 10,000 cells per spheroid and watched them through the span of 5 days. After many attempts, we concluded that the density of 5,000 cells per spheroid in the third day is the perfect size and perfect density to go through our tests. After the evolution in time, we wanted to do a microscopic fluorescent staining to see if the three structures of the spheroid are true. And so we used control spheroids, non-treated spheroids, and stain them with the fluorescent dyes like calcine AM, which is green and stains the live cells, and propidium iodide, which stains the dead cells and is red. And we can see that the live cells are on the outside, the proliferated zone, in which is true, and the dead cells are gathered in the center, which is the necrotic zone. After that step, we apply the treatment with gramicidin A, our antimicrobial peptide, and watch the effects on spheroid integrity. And we can see that at 24 and 48 hours, with the growth of concentration in antimicrobial peptide, we can see forming in the vicinity of the spheroid a cloud of cell residues, because apparently the gramicidin A has a puncture-like mechanism in which pokes hole in the membrane of the cells, and all the cytosolic organelles are leaving the cells and apparently dies. After all, the viability test of the cells using MTT and ATP test shows that the treatment is quite aggressive with the increase in concentration. The mitochondrial activity levels are lowering with the growth of the concentration, and also the ATP, I mean the energy levels are lowering with the concentration of the treatment. Well, after this, we also wanted to see the level of penetration of doxorubicin, because doxorubicin, a classical compound used in the chemotherapy, is fluorescent and also red. And so we wanted to see the level of penetration because it can interfere with our viability test, especially MTT test, which is an absorbance test, because doxorubicin makes the spheroids red. And we chose three different cellular densities and watched on the span of 4 hours, and we can see that with the passage of time the doxorubicin accumulates in the spheroid, making it more and more red and fluorescent. The effects on spheroid integrity are also aggressive, not so much visually, but we can see that the spheroid blackens, and which means that the cells inside the spheroid are dead with the increase of the concentration of doxorubicin. Also, we can see in the vicinity of the spheroid residues of dead cells. The test that we could perform because of the fluorescence of doxorubicin was the ATP test, which shows the energy inside the cells. And we can see that with the increase of concentration of doxorubicin, the ATP levels are falling, which gives a correlation with the viability of the cells. In conclusion, we had success in obtaining spheroids with similar characteristics as reported in literature, and the treatments applied to the spheroids were proven to be efficient. Our outlook is creating stable multicellular spheroids and study of a wide array of parameters like oxidative stress and protein or gene expression of proteins that are implicated in apoptosis and necrosis. You also using this multicellular three-dimensional system to study the effects of ionizing radiation, proton and X-ray. Thank you for your attention.
We want to thanks to Professor Mina Raileanu for her nice and very interesting presentation.